Good evening. Today we are going to learn about two important phenomena which are seen in fungi, that is heterothallism and parasexuality. In fungi, there are two types of sexual systems that have evolved, homothallism and heterothallism. Homothallism, when there is no difference between two thallus which are undergoing fertilization, and heterothallism when the two thalli which are undergoing fertilization, a successive compatible fertilization, have to be different. Hetero means different. So actually heterothallism can be compared to a dioecious condition in higher plants where male and female are different, and gametes coming from male and female, they undergo fertilization to give a zygote. Similarly, in heterothallism, two mating thalli have to be of different types to result into a compatible fertilization. They are found in higher fungi. In fungi, the thalli may be morphologically similar, but they are physiologically different, and a compatible fertilization only takes place between two physiologically different thalli. The hypha of two individuals are not differentiated morphologically at vegetative stage. At vegetative stage, we cannot identify that these two are of the same mating type or of different mating type. They only show this difference during their sexual behavior when two thallus of the same kind, they come across each other. If they are not undergoing fertilization, that means they are of the similar type, and if they are undergoing fertilization, that means they are of different type. So this behavior can only be seen during the sexual phase. It is the first indication of origin of dioecious condition in higher organisms. This phenomena was first of all observed by Blacksley in 1904 in Mucor. It's a member of Zygomycetes. Sexual reproduction, it was observed by Blacksley that sexual reproduction did not occur between hypha of similar strains, and only hypha of opposite strains, when they come across each other, it resulted into zygote. He identified them as plus and minus strain to differentiate from each other. Now homothallism means we can compare it with bisexuality, where both the organisms, both the plus and minus strains are within the same species. Mucor himalis and Allomyces exhibit this type of condition, whereas unisexuality in dioecious condition in higher plants can be compared to heterothallism. This is seen in Mucor mucido, Puccinia, Astilago-Medes. Heterothallism can be of different types, like Whitehouse in 1946 identified two types of heterothallism: morphological heterothallism and physiological heterothallism. Morphological, where by looking at the thallus we can identify that the two strains are different, whereas physiological, which are similar morphologically but exhibit a different kind of mating behavior. Now sexual compatibility is a genetic feature. Here we see that there are two types of heterothallism, bipolar and tetrapolar. Bipolar, when we are concerned about the two alleles of the same gene. The two mating groups, they differ by only one gene, and this gene has two alleles represented as capital A and small a. Only when the opposite type of alleles come across one another, they undergo mating. This is exhibited in members of Mucorales and Ascomycetes. Whereas tetrapolar heterothallism is exhibited by two genes, namely A and B, and these two genes have got their alleles in turn, like capital A and small a for the gene A, and capital B and small b for gene B, and diploid zygote has a genotype of AABB with four combinations as capital AB, capital A small b, small a capital B, and small a small b. This is exhibited by the members of Basidiomycetes. Now here in this picture we can see that mating type A and B, when they produce spores and spores germinate to give rise to a thallus, Mating type A will produce A thallus and mating type B will produce B thallus. A thallus will give rise to conidia or gametes of A type, and similarly B thallus will produce conidia and gametes of B type. Fertilization whenever occurs, it has to be between the conidia of two or gametes of A and B, and once successful compatible fertilization occurs, it results into fruiting body that is carrying again both A and B. Now then when this fruiting body gives rise to spores, again A spore will give rise to A thallus and B will give rise to B thallus. So this way, this is, we can, in a way we can say, it is a presumptive form of sexual reproduction seen in higher plants. Now our second topic is parasexuality. Parasexuality is actually a cycle, a sexual cycle in fungi where plasmogamy, karyogamy and meiosis, which are the three steps of a sexual cycle, are taking place, but not at a specified position or not at a specified time. They have means they undergo the process but in a different way, we can say. Okay, here true sexual cycle is lacking, but transferring of genetic material takes place without meiosis or without development of sexual structures. Plasmogamy, karyogamy and haploidization, they take place at random positions, and recombination takes place during mitosis. Otherwise mitosis is just a equational division, but here haplodization or recombination also takes place in mitosis. Now here, this was first of all studied by Pontecorvo and Draper in 1952 in members of Ascomycotina and Basidiomycotina. Formation of heterokaryotic mycelium takes place. The steps in parasexuality includes, first of all, formation of a heterokaryotic mycelium. This is similar to plasmogamy. Now this can take place by mutation. See, mutation can result into a heterokaryotic mycelium where change in genetic makeup results into a different type of a nucleus, and then same thallus is having two types of nuclei. So that is similar to plasmogamy. Or it can also take place by anastomosis between hyphae of different origin. When two hyphae of different origin, when they fertilize, when they come across each other, then this type of heterokaryotization takes place. Now homokaryons, when similar type of nuclei are present, like here we can say, we can see in the first instance that homokaryons from two different thalli, when they undergo a cytoplasmic fusion resulting into plasmogamy, so that the same thallus has got now two karyons from different thalli. Now this mixed haploid nuclei, which we call heterokaryons because they are coming from different types of thalli having different nucleus. Fusion of some of the nuclei results into a hybrid kind of a nucleus representing both the nuclei. This, when undergoes mitosis, some of the nuclei are of thallus 1, some of the nuclei are of thallus 2, and third type of nucleus are there which are a combination of 1 and 2. So this represents both the parental types and the parental genotypes as well as the recombinant genotypes. Now again, when this gives rise to a new thallus, the one having A type nucleus will give rise to A type thallus, B will give, second type will give to second, and the third, the recombination will give to another type of thallus. Now fusion between two nuclei take place to form heterozygous diploid nucleus. The three type of nucleus formed are haploid, diploid homozygous and diploid heterozygous. Haploid, the ones that have not undergone recombination, and if they have undergone karyogamy, then karyogamy from the similar type of nuclei, they result into diploid homozygous. And if karyogamy takes place between two different type of nuclei, it results into diploid heterozygous. Multiplication of diploid nucleus takes place by mitosis. Here only mitosis is the division that results into multiplication, and since there are certain heterozygotes available, so they will give rise to new heterozygotes. Occasional mitotic crossing over takes place. This is the most important step of parasexual cycle, because crossing over is not an event that is normally occurring in mitosis. Crossing over is an event that is occurring in meiosis, that is meiosis 1, prophase 1. That is the important event that is occurring in meiosis. But here parasexual cycle, in parasexual cycle, crossing over takes place in mitotic division, and this leads to genetic recombination, and in spite of having absence of meiosis, the benefit of sexual cycle are achieved in this also. Then the diploid nuclei, they get sorted, and conidia that are developing, they are the type of conidia that will be on the basis of what type of nucleus is obtained by the thallus. Sometimes haplodization of diploid nuclei also takes place, and this results into again development of haploid conidia. New haploid strains are again sorted, and now different haploid mycelia which are developed from haploid conidia are different in genotype from the mother mycelium, as they have been developed from mitotic recombination. This is the parasexual cycle. Here we can see that the anastomosing between two type of strains results into heterokaryon. Then heterokaryon, it undergoes diploid dikaryoticization. Dikaryoticization then results into haploid nuclei, as well as the karyogamy resulting into a heterozygote. Now this heterozygote, if it is giving rise to a new thallus by mitosis, then heterozygotes will be produced, or if haploid are giving rise to thallus, a haploid thallus will be produced. So this type. Now in the next part of the cycle, when haploids they come across each other, they will give rise to a plasmogamy, karyogamy will give rise to a diploid individual, and the diploid heterozygous, it will only undergo mitosis to give rise to a diploid heterozygote. So diploid heterozygote is obtained either by crossing over fertilization between the two haploid thalli, or by multiplication of the diploid heterozygote. So this way parasexuality means a sexual cycle that is actually not existing in the form that is present in most of the species, but it is actually in the form of in a different form, where plasmogamy, karyogamy occur, meiosis may or may not occur, but alternation of generation takes place. So these are the two phenomena that we come across while studying fungi, and this way fungi, they exhibit a different kind of sexual cycles, parasexual cycle, which is not seen in higher plants. Thank you.